Hey, hello everyone. Welcome to the data showcase. So every year at NeuroHackademy, we have one of these data showcases where we basically just take some of our favorite data sets that are available online and for you to use that make good data sets for hack projects next week. And we show you a little bit about them and give you some examples of how to download them and use them. So basically, we're going to walk through a bunch of data sets today. And we're just going to do quick little examples of how you can interact with each of them and maybe show you some plots or something like that. In general, if you have more questions about these data sets, all of the data sets we're going to present today, we are prepared to help you with next week. If you've got some other data set you'd like to know about, we can't promise that we'll know about it, but we'll do our best. But for sure, if you want to work on one of these, we can help you. However, just to get started, I want to introduce a couple concepts that we may not have seen in the course so far. And the first one is we're going to talk a little bit about the file system in Python and paths. So some of you may be familiar with the Python OS package. It's pretty commonly used. It's a way of operating with the operating system, of interacting with the operating system. And there's a whole submodule of OS called os.path, which is for dealing with the file system specifically. And part of the reason you might want to use a module like os.path is because you don't know if your code's going to be run on Windows or on Linux, and you don't know if you need to use backslashes or forward slashes to separate your directories. And so you might want to use the os.path.join method or function, which can take part of a path and a subpath and join them together with slashes or backslashes, depending on your operating system. This is a perfectly fine way to do Python code, and it's probably, if you look at a lot of Python code, you'll find these kinds of calls all over the place anytime you interact with the file system. However, there's another library called pathlib. I don't remember when pathlib was introduced to Python. It's probably pretty old, but it makes operating with the file system a little bit easier. You can, so here's some example code. I apologize, I'm not going to live code through this session because it's got a little bit too much for me to remember. Here's some example code. We can import the path type from pathlib, and then we can just declare a base path as this particular path. This is our home directory on the hub here. And then we can declare a subdir, and we can combine them by using what looks like the division operator, but basically, since slash is the character you use on Linux and Mac OS to separate directories, they use slash or division as like the operator that you can use to join paths. So this creates a representation of the curriculum directory in the home directory. Does that make sense? Any questions about this? Okay, so let's just look at a couple examples real quick. We're not going to spend very much time on this. But here I'm actually importing path from pathlib, and then I'm making a cache path in the /temp directory. I'll talk a little bit more about cache paths in just a second. And then here I just have a line of code that says, if this path doesn't exist on the file system, go ahead and make the directory. And that ensures that we have this directory exists on the file system. Okay, so far so good? Okay. Here we have another example where I take that cache path that we just made. I'm going to run this. I take that cache path we've just made, and I get the file readme.md inside of that cache path. And this is basically, I'm just going to make a readme file with a few lines that say that this is a cache path, and this directory is safe to delete because it just contains cache data. So here we can say, we can make this readme file. We can open it in write mode, just like you would use the open function in Python to open a file. And then we can print a few lines to the file. And I'm just going to check that it worked correctly. If we run this cat command, we'll see the contents of that file right here. So this is just to, in case you're not familiar with these kinds of pathlibs and these file operations, I just wanted you to know about them, because we're now going to move into using a library called cloud pathlib, which is basically like pathlib, except it works with things up in the cloud. And it's a really nice little library. I forgot I had another example here. If we wanted to define an ls function to list the contents of a directory, we could write it very quickly. It would just be if the path is not a directory, we raise an error because we can't list a non-directory. Otherwise, there's a method of paths called iterdir that will give you all the contents of the directory, and we can just return that as a list. So we can run that, and you see that we get back a bunch of paths that represent the contents of this directory. You probably don't get the same results if you run it because you haven't run this notebook already, whereas I have. I already cached all the data, so I wouldn't have to wait while it loads. And so I've got a bunch of other directories here, but you should probably just see this README file if you run it. Yeah.
How do you handle these objects? Objects, is it like just a string or?
Yeah, so the POSIX paths objects are the same thing as the path object. It's just a particular instance of a path object, and basically you can convert it into a string if you have a path. So, for example, let me just get a new cell here. If we look at the cache path, it's also a POSIX path, and we can just ask for the name, and that'll give us cache. We can convert it to a string by calling the string function, and it'll give us back the file name. But it's also got a bunch of methods on it that are very handy for interacting with the file system. Yeah. Other questions? Okay. Okay. So CloudPathLib is basically a library that provides a few kinds of path-like objects, but they work on paths up in the cloud. So in particular, we're going to go through a bunch of data sets today, and I think all but one of them live on Amazon S3, which is Amazon's storage solution. And basically in S3, you can have S3 buckets, which are sort of like a directory in the S3 root, or the main S3 kind of space. And you just have like a file system inside of these buckets. And so you can have a path that starts with S3://, and then you have the bucket, and then you have a slash, and you can just reference files inside of that S3 bucket. And we'll see some examples of that very shortly. But if we want to create one of these paths, what I'm going to do here is I'm going to start by importing from CloudPathLib, import S3 path and S3 client. Now, S3 client, we have to import because in order to talk to these paths in the cloud, we need to do some kind of authentication with Amazon. Typically, for a lot of these data sets, the authentication is very minimal. They're not really tracking who's logging in, but you still have to have some kind of logic that talks to Amazon and performs those kinds of functions. So we can make a client here, and we just provide two arguments. The first one is the local directory where we want these paths to cache the data. So the thought here is that all these data live up in the Amazon S3 in the cloud. And in theory, you could just take a file and you could read it straight from the cloud, and that would be fine. But in practice, you often want to access the same file multiple times in a single session. And you may want to do other things with the file besides just read it as a stream. And so it's often easier for the library to download the file, put it somewhere on your local file system as in like a cache directory, and then let you read that file directly as if it were a normal file. This is especially useful because a lot of libraries like NeuroHackademy don't necessarily operate on cloud paths. You can ask NeuroHackademy to load an S3 directory, but it doesn't necessarily know how to load an S3 directory. But if you give it a file on your local computer, it can always load it. So by having a cache directory, we can make sure that our files get cached locally on our hard drive. And then the second option is no sign request. It basically just says we don't need to log in order to access the data set we're going to be looking at. We'll look later at the Human Connectome project where you do need to log in. And if you're hoping to work on the Human Connectome project, I posted some instructions for how to get credentials. If you haven't followed those instructions, the HCP examples in this directory won't work. You'll have to have done those first. Okay, so just as a quick example, the natural scenes data set, which we'll talk about in just a little bit, is one of the data sets that is kind of a nice data set for the hack projects next week. And it lives in a bucket that's just called natural scenes data set on S3. And so the path is just S3://, kind of like HTTP, natural scenes data set like that. And we give the S3 path the client that we just created. And this will give us back, and I'll actually add this at the end here so that we can see it. an S3 path object representing that the roots of the bucket for the natural scenes data set. So we wrote this ls function up here that was supposed to work with pathlib paths, but it'll also work with cloud pathlibs because cloud paths. It'll work with them because the cloud paths basically act just like paths. They're not paths, they're not local paths, but they're paths in the cloud, so they behave very similarly. So we can go ahead and just run this ls command, and it'll give us back a list of the subdirectories in the natural scenes data set, and files in this directory. Okay, any questions on this so far? All right, so that was just an introduction to paths, just because we were going to be using this cloud pathlib to access some of these data sets. So I wanted to make sure that everyone was familiar with that. So with that, let's go ahead and talk about the Human Connectome Project. So you've probably all heard of the Human Connectome Project, I'm guessing. It's been around since at least 2012. It began in 2009 as an NIH Blueprint Grand Challenge. It actually went through several stages and was there were multiple consortia that worked on this project. It initially started with a bunch of efforts to develop MRI sequences in order to be able to have really high quality MRI data at a bunch of different centers. And that was part of one arm of the Human Connectome Project. It's not the arm we're going to talk about very much today, because we're more interested in the MRI data collection arm. And that's called the Young Adult Human Connectome Project. So the Young Adult HCP was largely spearheaded by David Van Essen and Camille Ugerbill in kind of joint venture with the Washington University in St. Louis and the University of Minnesota. And there have been quite a few follow-ups and additions to the HCP. So I don't know if they're still running HCP data collection grants, but for a long time there were grants you could get to collect like low vision subjects for part of the HCP, or across the lifespan kind of data collection efforts that various labs could participate in. They have a single facility where they coordinate all of their data, and it is humanconnectome.org. And we're just going to visit that real quick, just so you can kind of see it. This is basically their main website. And in particular, if you go to not humanconnectome.org, but db.humanconnectome.org, I believe, it'll take you to their database access page. I'm going to make this a little bigger. Now, if you follow those instructions that I posted, you've already seen this, because you have to log in. You have to register for an account and log in. And then once you've registered, they will just give you credentials that you can use to access the Human Connectome Project. So it's not like you have to pay for it or anything. It's just that they want you to register in order to be able to use the data. I'm not going to log in here, but I will just tell you a little bit about this website. Once you've made an account, you can log into this website and you can actually download their data on the web if you want. It tends to arrive in great big, like many gigabyte zip files, and it can take quite a while to download. And in particular, there's a lot of pre-processed metadata in those files that you may or may not need. And so if you have a particular analysis goal, it's often easier, at least in my experience, it's easier to download it from S3, because that way you don't have to download all the extra stuff. As an example, I generally don't care very much about the raw data when I'm doing my analyses. I want the pre-processed data. The raw data can be very large, and I don't really want to download it all. I just want the pre-processed data. So you can have a little more control over what you get when you use these libraries like the Cloud Pathlib to access it. Real quick, I just want to mention that you can also use the AWS command line if you're familiar with it. There's a command line you can get from Amazon Web Services where you can basically use a command like AWS S3 copy, and then you can give it an S3 path and you can copy it to your local directory. That's a perfectly good way to interact with the Human Connectome Project. I'm just going to show you how to do it in a Python notebook though. Okay, so the Young Adult Human Connectome Project, I think they released most of their data around 2012. I don't remember the exact timing. But they have scanned about 1,200 subjects with 70 MRI for about 200 of the subjects. And they collected a whole bunch of data. They had structural data, they had diffusion weighted data, they had various functional tasks, including some working memory tasks that were done in the scanner. For about 200 of the subjects, they collected retinotopic mapping experiments, which is something I care about a lot because I study vision. They also collected various behavioral data, including things like vision tests and various clinical assessments. And they got some genetic data. So interestingly enough, a whole bunch of the subjects in the Human Connectome Project are twins. And they have a very big set of both monozygotic and dizygotic twins. So if you're interested in genetics and heritability, this is a very interesting data set just for those reasons. They extensively pre-processed these data, and they kind of invented their own processing pipeline just for the processing. It's called the HCP pipelines. Oh, whoops, didn't mean to do that. So you can find the HCP pipelines on GitHub. Essentially the HCP pipelines, if you're familiar with FreeSurfer, they basically start by processing each subject with FreeSurfer. They then take the outputs of the FreeSurfer processing and they do a bunch of extra stuff to it mostly to make it easier to compare left and right hemisphere across subjects. So in FreeSurfer, if you run subjects through FreeSurfer, there isn't a correspondence between right and left that you can use to easily compare one anatomical position to another anatomical position. FreeSurfer makes it relatively easy to compare across subjects as long as you stay within the same hemisphere, but comparing right and left is kind of hard. The HCP pipelines essentially take FreeSurfer and also give you a left and right hemisphere alignment so that you have approximately equivalent anatomical positions on the left and right hemisphere you can compare. That's my very high level overview of what the HCP pipelines does, but it's actually fairly complicated and does a lot of stuff using some of their own algorithms that are worth looking into if you're interested in these kinds of topics. Okay, so I have included the instructions in this notebook for setting up the HCP credentials. I'm not going to go over them right now because it would involve me dumping my password and stuff into the Zoom, so I won't do that. I hope I hope that anyone who wants to follow along has already done this. But if you haven't followed those instructions I put in Slack, then this next couple things will not work. You'll just have to trust me. Okay, so basically, we're going to, in order to access the Human Connectome project, we're going to start by importing pathlib and importing S3 path and S3 client from Cloud Pathlib. I'm going to go ahead and set up a cache path here, and I'm putting it in /temp/cache. And then we just make sure it exists. Then to create the S3 path, we just pass it S3://HCP-open access. So this is the name of the S3 bucket for the Human Connectome project, HCP-open access. And HCP_1200 is basically the directory that contains the young adult 1200 subject release of the data from the Human Connectome project. So this is just a path to their kind of base young adult Human Connectome project release directory. We provide the S3 client with that cache path that we made. And finally, we provide a profile name. And that profile name refers to the profile in your AWS credentials file, which is where you put the Human Connectome project credentials. Again, if you didn't see the instructions I posted earlier, that may not make sense, but the instructions up here explain to you how to set that up so that you can use that profile. And that basically just tells it how to authenticate as me. So I'm going to go ahead and run this. And go ahead and make sure that this returns something reasonable. We can see that it's given back this path. Okay, so let's go ahead and look at what's in that path. And to do that, we could either run that ls function that I wrote, but here I've just written it as iterate over the directory and make a list. So this is going to give us back a list of all the paths that are subpaths of this HCP 1200 directory in the cloud. And there are 1200 subjects, or about 1200 subjects in the HCP, so I'm not going to list all of the directories. I'm just going to list the first 10. which is what I do here. And you can see that these are the first 10 directories. Basically, all of the subjects in the Human Connectome project have 6-digit IDs. The very first one here is 1000206, etc. So these are all different directories, one for each subject. Now you'll notice this is not in BIDS format. There are, in fact, I think most of the data sets we're going to go over are not in BIDS format. Part of that is because some of these are old. The Human Connectome project kind of came out a little before BIDS came out, I think. So I believe they just didn't know about it yet. But regardless, there is a kind of standard directory structure for the Human Connectome project that we'll look at a little bit here. So now, just real quick, I'll mention that there's this utils.py file here in this directory. We put a few just like utility functions in there. And one of them is called crawl, and it'll take a path or a cloud path and it'll just crawl over it and print all of the subfiles and subdirectories to the screen. So we're going to do that, but we don't want to crawl over this whole directory because it's massive and it would take forever. So instead, we're going to take one subjects directory. So we'll take 100610, which I think is this subject right here. This subject is interesting mostly because they have a very big brain. Just interestingly enough, they were the biggest brain of the 200 subjects that did 7T Renatopy in the Human Connectome project. We're going to look specifically at a subdirectory in their subject directory called T1W for T1 weighted, and then another subdirectory named after them. And that just happens to be the place where the Human Connectome project stores the free surfer output for this subject. Now, one of the things about the Human Connectome project that people often complain about is that their directory structure for Human Connectome project subjects is a little unintuitive, and it contains duplicates of files, and it's very difficult to kind of come in at first glance and understand how the files are organized and what each one means. Some of us have spent a lot of time mucking around in the Human Connectome project directory, so if you are interested in doing a project on this, we can help you. But you'll just have to take my word for it now that this contains the free surfer data for this particular subject. And basically, we're just going to make a cloud path object that represents that directory, and then we're going to crawl it and see what it outputs. So basically, if you're familiar with free surfer's output, you'll recognize this as free surfer data. But we have the base directory, then we have label, and then we have a bunch of label files and threshold files, which is what free surfer outputs in the label directory. We come down here to the MRI directory where we see a bunch of various transformations, and then also there should be some number of these like MGZ files, which are free surfer's version of Nifty files, more or less. So we have one for like the T1 and various segmentations and the brain mask and all this kind of stuff. Then there's the stats directory, which, you know, as much as I've used free surfer, I'm not even sure what the stats directory contains. And then we have this surf directory, which contains all of the models of the cortical surfaces, et cetera, et cetera. So you can see that all these data can be pretty easily crawled and pretty easily accessed using this cloud path lib. Now, if we wanted to load one of these objects, we would need to use NeuroHackademy because NeuroHackademy understands what's in the file formats, or understands the file formats for these files. So here I'm just going to import NeuroHackademy, and I'm going to get the path for this the brain.mgz file. And just to kind of drive this point home, I want to go ahead and show you that this is an S3 path. This isn't an actual local file system path. This still is just something up in the cloud. But we can ask an S3 path for the file system path, and it will make sure that that file is cached and then give us back the path on our local cache directory. And that's something that NeuroHackademy can load. So once we found the file we want, we can just tell NeuroHackademy to load it from the file system. And that will take maybe a little bit longer for you to run if you're following along, because for you have to download it. I did this earlier, so it's already cached. But it shouldn't take too long. And then here I'm just going to import matplotlib and go ahead and plot an arbitrary slice out of the middle of this image that we just loaded in. And it should look like a brain. Yep. So there we have that subject's brain that we just pulled down from S3. And I'll just mention as well that there's one other thing, which is that suppose we had something like this brain image path. There's also a function called download to, and you can give it a path where you want this file to be downloaded to. And I believe it works on directories too. So if you want to download a subject directory and you want to get all of the data, you can go ahead and tell it to download to a particular path. I think this is probably a good time to say that if you are planning to work on a data set like the Human Connectome project next week, please don't just download everything into your home directory. You will have limits. There's a limit on how much data we can have in your home directories, but we can put data in the shared directories for you. So if you want to work on the Human Connectome project, let us know what you think you want, and we'll download it to the shared directory. So to a large extent, you won't have to worry about that. However, I find these kinds of libraries, the cloud path, I find these great for working on in my own workflow, like not when I have a Jupyter Hub, but when I'm just working on my computer, because you can explore the data without having to download it all before operating on it. Questions so far? Okay. So, going to go on to the Human Connectome Project 7 Tesla retinotopy dataset. I won't spend too much time on this, but this was a project that I did with Jonathan Winwer and Kendrick Kay a few years ago. Basically, the Human Connectome Project, and by the way, I'm not involved with the Human Connectome Project directly, I just do analysis of it. But the Human Connectome Project collected a bunch of retinotopic mapping data for vision, and they published the bold data, but they didn't try to turn those data into models of the visual system or anything. So we basically took the bold data and modeled it as population receptive field models and then published those models. I'm not going to take a deep dive into vision right now, but if you're interested, ask me later. But I did want to show you, so there's a library I maintain called NeuroPython that also has the ability to access a lot of these libraries kind of built in. So here I'm just going to kind of demonstrate that real quick. We're going to import NeuroPython as NY. There's a little function that we have in the utilities for loading our AWS credentials. And we basically just tell NeuroPython through its configuration interface that we have a set of credentials and that we want things to be cached in this temp directory. We'll go ahead and run that. And then we can just ask NeuroPython to give us back a HCP subject. And the thing that NeuroPython does, in fact, NeuroPython was originally written to make sense of the output of FreeSurfer. FreeSurfer produces all these files and it has command line utilities for interacting with them. But oftentimes you just want that data in Python. And that's kind of what NeuroPython does. And when the Human Connectome project came out, I just extended it to also understand the Human Connectome project data structure. So we can just ask it for a Human Connectome project subject. It knows where to get it, and it's going to give us back an object. And if we want to make a plot of this subject's cortex, we can use NeuroPython's cortex plot function, tell it we want to plot the left and right hemisphere, and we're going to show the white surface. Hopefully this works. This may take a second, especially for if you haven't downloaded the subject already, it'll need to download just enough data to be able to make this plot, basically. And it probably needs to load the 3D rendering library in my case as well. Questions while this loads? Okay. Well, there we go. Yeah, so you need to set up the credentials, you need to get a few lines of text into that .aws/credentials file. And the easiest way is just to dump them in a terminal. But if you want to use an editor, that's fine too. When you tried to load it, didn't work. Okay, well, sorry about that. I can take a look at it in a little bit here. Yeah.
Is there external software needed for the visualization?
So this, yeah, great question. For NeuroPython, it uses internally it uses a library called IPyVolume, which actually has a JavaScript interface in the Jupyter browser. So typically you can just install IPyVolume and it'll render these in your Jupyter notebook without much trouble. Yeah, I've not it used to be a lot harder to get these to work, but lately the installation has gotten pretty smooth and these work pretty well. So yeah, so it renders a fully interactable 3D model in your browser. It's a great way to look at brains. It's a great way to, you can zoom in and see how we actually model these brains is by a bunch of little triangles. Anyway, this may be a review to a lot of you, but I just wanted to demonstrate that. Now, NeuroPython can, this cortex plot is fairly sophisticated. Here I'm just doing an example with the inflated brain, and I'm asking it to color the V1, which is first visual, cortical visual area. I'm asking it to color it red. It knows about the V1 label because the V1 label is standard FreeSurfer data that FreeSurfer calculates, and that comes along for the ride when you load in the subject. So this will take a second to render as well because it needs to download these inflated cortices, but there it is. Yeah, so there's the inflated cortices with V1 in red. Okay, so the red and toppy dataset exists on the Open Science Framework on osf.io, and I do have a link to it in this notebook. But really, I would encourage you to interact with it through NeuroPython if you're going to interact with it. It just makes it a little easier. And I just have a quick example here to show where basically we take this subject, we're going to color it by the polar angle, and I'll explain what that is in just a second. And we're going to mask the polar angle to just where we're able to explain at least 10% of the bold signal in terms of our visual model. So this is basically just saying we're only going to plot polar angle where we're confident that the cortex is visually responsive. And we're going to plot it on the inflated brain. So that'll take a second. This may actually take longer than anything else just because it needs to download the Renatopy data set. But while it downloads, I'm just going to explain real quick what polar angle is. So this is a legend of polar angle, and the circle represents your visual field. And we're assuming that you're fixating, like your eyes are pointed at the very center of the visual field. Then this image is colored according to the polar angle. Polar angle is just rotation around the visual field. And it's orthogonal to eccentricity, which is the distance from your center of gaze. And the idea is that like as you look around, your center of gaze is always changing, but your polar angle is just the rotation around it. Okay, this didn't take that long to load. So here's the polar angle plotted on that subject. And you can see that there's this very clear kind of pattern of polar angle right around V1, which is the first visual area. And that's Renatopy. Okay, questions on any of this so far? All right, Ariel, you want to do the next one?
Okay, so still in the Human Connectome project, as Neil mentioned, the Human Connectome project collected several different experiments with every one of the subjects in the data set. And one of the experiments that they did was to collect diffusion MRI. Here's the pictures of diffusion MRI. It's not from the Human Connectome project, but it demonstrates the kind of things you can do with diffusion MRI. Diffusion MRI sensitizes the MRI measurements to a particular location, but also a particular direction of diffusion. And so using that information measured in multiple different directions, you can make inferences about things like the mean diffusivity, which is the map here on the left. This is a single slice, horizontal slice through a person's brain, and mean diffusivity is relatively low in parts of the tissue where there's lots of tissue restricting the motion, the diffusion, random diffusion of water. This in the middle is a map of so-called fractional anisotropy. This is a measure of how anisotropic diffusion is. That is how much more it diffuses in some directions relative to other directions. And that value is pretty high in parts of the brain that contain large bundles of white matter that restrict the diffusion of water across their boundaries, but allow diffusion along the length of those bundles. And then finally, this color-coded map here is called principal diffusion direction. It's encoded through R, G, and B for right, left, anterior, posterior, and inferior, superior. So for example, R encodes the right, left direction. You can see the corpus callosum, the principal diffusion direction, the direction along which there's most of this diffusion, relatively speaking, is the right, left direction because it's right in the corpus callosum. In contrast, if you look right here, it's blue, which encodes the inferior superior, and that's roughly where the corticospinal tract goes from our cortex down to the spine. And so that's where there's a big bundle where water can diffuse more freely up and down. The experiment that the Human Connectome Project did produced pretty fabulous diffusion data, even though this was about 10 years ago when they collected this data. I'd say that the acquisition that they used, the processing methods they used are still really state-of-the-art. I have a hard time pointing to a data set that has diffusion data that is more of higher quality, that allows more different kinds of analysis than the Human Connectome Project. It's collected at 1.25 millimeter isotropic voxels. They collected 270 different volumes or directions in every subject, and 3 different diffusion weightings. The multiple diffusion weightings open up the possibility for all kinds of modeling that is hard to do when there's only one diffusion weighting. So data is really quite remarkable. And now I'll show you how to get it. So we're going to use a very similar kind of approach to what you've seen so far. So this is the first way of getting the data. I'll show another way of getting the data in just a second. So far, very similar to what you've seen so far in Noah, the notebooks that Noah walked us through. Within this T1 weighted directory, instead of putting in the subject's ID here to get the free surfer folder, instead I put in T1 weighted diffusion, and that's where the diffusion data is hiding within the folder structure of the Human Connectome Project. So you can see this file called bvals encodes the diffusion weighting values. This one called bvex encodes the directions of diffusion. And this data.nii.gz encodes the data. There's also some other things here, like a brain mask and so on. And so this is the path to the diffusion data for this one person. This might take a little while because it's probably caching it locally. And I won't even go into doing this because this requires a lot of memory. So the data, I believe each one of these diffusion files is about 4 gigs of data. And it'll actually break the hub if we do that, because we have a limit of 8 gigs here, and apparently it gets duplicated when NeBabel reads it. So you're asking, okay, what can we do if we're working on the hub? So 2 things about that. One is that we've requested to increase the memory that each one of us has to 16 gigs starting on Monday, since we know that you'll all start working with lots of data on Monday. So hopefully that will give us a little bit more breathing room. But there's also alternatives. I'll show a couple of alternatives, but before I go into that, I'll show a different way of grabbing the diffusion data. So as Noah pointed out, the data here is not organized according to bids. But for our own work, we needed to have data in bids so that we can run pipelines that rely on bids on this data. So we developed within the DiPy library, we developed what we call a fetcher. So many libraries, you've seen this this week, I guess, from NeLearn and other libraries, have little functions that fetch some data that you want to fetch, usually for examples and things of that sort. In DiPy, we have a set of fetchers as well. And a lot of them are used in the examples that are part of the documentation of DiPy. I should say a word about DiPy. DiPy is diffusion imaging in Python. It's a library that implements many different methods for analysis of diffusion MRI and computational neuroanatomy more generally. It's been around we've been developing it for more than 10 years now, and is used by many groups to do a lot of different things. And we use it as the basis for methods that we develop for tractometry. And I'll talk about tractometry in a minute here. But first, I'll show you that you can fetch the Human Connectome Project data using this kind of fetcher. So here, we give it a list of subject IDs. For example, this very first subject. And that now it's downloading it, and it's downloading it into a directory in the home directory, and actually Noah's home directory here. .dypy, that's where dypy stores its data. Inside was something called HTTP 1200, derivatives, HTTP pipeline. So we're pretending that HTTP is like a BIDS data set, and we call it HTTP pipeline and derivatives because those of you who were in the BIDS session know that derivative data sets, not the raw data, are stored in derivatives folders. In this case, this is not the raw data. What we're downloading here is the data that's been pre-processed by the HTTP and using their pipelines. And that's why we're storing it inside of this HTTP pipeline. And so everything, all the B values and BVEX go in there and get names that are BIDS-ish in nature. Let's see if this works. And then we can interact with this thing as though it's a BIDS data set. It is now a BIDS data set, and we can interact with it as though it's a BIDS data set and get different aspects of the data and work from there with the BIDS toolchain. Okay, let me actually pause here. What questions do people have so far about this? Yes.
Well, if you had that subject ID, how would I know kind of how to start accessing it?
Right, where do you get the subject IDs from? There's somewhere there is a where is it? There's a 12 like a NeuroHackademy has a list of these IDs actually stored in a variable, right?
Yeah, and actually we just OS the HTTP_1200 directory, the sub directories of that are
Okay, inside of cache, temp cache.
oh, I think it's in /HTTP. No, sorry, /temp/cache/HTTP. But I was thinking LS the
Oh, I see. LS. This one.
/HTTP 1200. It's literally real life somewhere.
Ah, yes. Ah, your computer is not my computer.
Yeah, we'll take it.
There, and in here, there's a text file, I believe. Oh, well, this is all the subject IDs, right? So you got them right there. I think there's also a text file somewhere in there. And NeuroHackademy also has, I believe, but I don't remember exactly where I
I
imported.
believe it's neuropython.http.subject_list.
Oh, it's downloading it from somewhere. No, it's in here somewhere.
I swear it's there.
It's in there somewhere.
This is how you actually work.
Subject IDs. There you go. There's a list there of these IDs, and you can start downloading. Oops. Okay. Yes.
So for NeuroHackademy, there's also a list of subjects in NeuroHackademy that have NeuroHackademy, and when I get back up, I can find it.
Yeah, so the question was, how do we find subjects in the HTTP that have both NeuroHackademy and also say diffusion? Because not everyone has diffusion. Actually, there's only 1,041 subjects who have diffusion, presumably because the rest of the data was not of sufficient quality or something. So there were some problems there.
So I think that all the NeuroHackademy subjects also have diffusion. I think that's the case.
No, not really. There's like one or two that don't have diffusion. So there's.
So I would, if it were me, I would just rather scrape that and check for that diffusion.
Yeah. Yeah. Yeah, I mean, we've done these kinds of things where we want to do diffusion, like some analysis of only the red entropy subjects, and my empirical way was just go through the entire list and just let it fail when there's no diffusion data. So I don't even know, I can't even tell you which is that subject that didn't have the diffusion. I don't remember. Okay, what other questions do people have? Okay, but we can go a little further here. A lot of analysis of diffusion data takes a long time, is cumbersome, is tricky to set up, especially when the data is this massive. And so if you want to work with the results of the analysis and do some kind of analysis of resulting data, for example, cryptography on the HCP or some characterization of the tissue properties within this, or you're interested in a particular part of the anatomy, good news for you, and that is that we've done a lot of analysis with the HCP and we've now put it on AWS where you can access it. So in my group, we focus on a very particular kind of analysis called tractometry. Tractometry, roughly speaking, this is a figure from a paper by Jason Yateman in 2012. Roughly speaking, we focus on delineating large anatomical structures within the white matter bundles that are well known, well recognizable in every individual that has a healthy brain. And we try to find those, and we try to find them in every individual. And then when we find them, we characterize tissue properties like fractional anisotropy along the length of that bundle. So these bundles have some interesting properties that the tissue properties like fractional anisotropy are not uniform throughout the length. You can see here what we call a track profile along the length of the corticospinal tract, average across multiple individuals. So first of all, there's variability between individuals, which is interesting because this variability could relate to some individual differences in the way that their brains work in all kinds of ways. And the second thing is also that there's systematic variance across the length of the bundle. So this tractometry approach captures the systematic variance along the length of the bundle, in addition to sort of differences between individuals. And there's a lot of things that you can do once you have that information. Along the way, we also do a whole brain tractography of every individual in this data set. So if you're interested in doing something else that's derived from tractography, like calculate connectivity matrices, that's also something that you can do with this data without having to, the main kind of bottleneck computationally here is the tractography. Generating all these streamlines takes hours with this data set. So we use this pipeline called PyFQ and ran it. Here are track profiles, average track profiles for 24 bundles, averaged on for all the subjects in all 1,041 subjects who have diffusion data in the HCP data set. And we put this up on AWS on the Open Neuro data. This is really confusing because it's not Open Neuro, it's Open-Neuro data. Different project that collects actually a large variety of different kinds of neuroscience data sets. This is run out of Joshua Vogelstein's group at Johns Hopkins University, and they have sort of this open connectomes project. These are connectomes from various different organisms, everything from worms and flies to humans and other mammals. And so we're putting that there together with these other connectomes. There's actually another set of data from the diffusion that Jovo's lab also processed using a different pipeline that also depends on DiPi. So you can compare our tractography with their tractography. It uses very similar methods, so it shouldn't be very different. There's a whole lot of data in there. So I'm going to focus specifically on this, on our tractometry derivatives, and just show you where you can find things in there, and then you can explore that further. So I'm using a very similar approach to before. It's another S3 address, but apart from that, these calls are very similar to what you've seen before. And then I'm going to crawl the derivatives path just for this one subject. Notice that it's now named in a BIDS-ish manner, and the file names here will be roughly BIDS-ish, although they're not fully BIDS compliant, unfortunately. The first thing to notice here, this is a folder with ROIs that we used along the way. It's actually not very interesting These ROIs are not so interesting. They're visualizations of different bundles that we found. And then there's a whole bunch of stuff in here, including different models of diffusion that we fit to this, different tissue properties like FA, the fractional anisotropy, mean diffusivity, mean kurtosis, because we used a model that took advantage of these multiple diffusion weightings to estimate the kurtosis of the diffusion. What else do we have in here? We have something called axonal water fraction, which uses this kurtosis in order to estimate the axonal water fraction within voxels, sort of a microstructure model of diffusion within the voxels. And then we have the tractography in here. We have and various other things. One thing, if you're interested, well, a lot of different things. And here's the whole brain tractography. So there's the tractography after it's been segmented into these different bundles that contains only the streamlines that belong to one of these bundles. But this one is a whole brain probabilistic tractography that includes all the tractography. So one thing you can do here is using pandas. If you know where to find for this subject, this file called afqprofiles.csv, and the only thing that will differ between subjects is this string here. So you can get this for all the subjects. If you know where to find that, then you can give pandas the cloud path here, and pandas knows how to read from AWS, and it will read that CSV file directly from AWS. Yeah, pandas knows how to take S3 addresses and read CSVs. If those CSVs are accessible, it'll just read it from there without having to copy, which is, yeah, it's kind of great. And then doing some more pandas operations here just to demonstrate. Yes, we got the FA. Here's the FA of the single individual along the length of their left corticospinal tract.
I guess that's basically the speed that's more
this property of pandas that it can read from S3. JB is asking, is this specific to S3 or is it just generally for like stuff that's online? And yeah, if I gave it also an HTTPS URL that has a CSV file, it would do the same thing. Yeah, it's not specific to the cloud path library. If I gave it, yeah, if instead of this cloud path, I gave it an S3 address, the S3 address of this thing, then it would read it just as well. I can give it HTTPS addresses. I could give it, I think, addresses on Google's storage, gs:// also works. Yeah, pandas has a lot of magic under its hood.
I suspect it turns the cloud. I suspect it calls the string function on the cloud.
Yeah, and then this I'll just try to translate what Noah just said into Zoom, which is that there's probably various ways that it does it here, and it finds the right way and it does that. This is nice because if like going back maybe to some stuff we talked about in the machine learning session, where like you often work really hard to do feature engineering and do something you can do machine learning with. These are features you can. These are highly engineered features. We started off with like a whole 4 gigabytes of data of an individual, and we ended up with a table that is, I don't know what's the shape here. It's 2,400 rows with 7 columns for each subject. So it's really reduced down, and we think these are really good features. We have some empirical evidence that they're pretty good features. Yeah.
So this might be an question.
Yeah.
Is there a way to, like for cortical voxels, be able to assess like what tracks they're likely receiving the majority of information from, or areas of cortical voxels that might be connected via tracks?
Right. The question is there some way, given this cryptography, to make inferences about where in cortex this connects and what different parts of cortex? So this is, I'd say, an area of active research. We're doing a little bit of this kind of research, connecting, for example, the kind of maps, random topic maps that Noah is just showing on cortex with these kind of bundles. Do the boundaries between different bundles tell you where the boundaries are between these maps? And the answer is possibly yes. So it's a really interesting topic of research, and not fully understood yet. Okay, let me pass this back to Noah for NSD.
Okay, so the natural scenes data set is a kind of interesting data set that was largely pioneered by Kendrick Kay at the University of Minnesota. Kendrick has been a common participant here at the NeuroHackademy, so we've used this data set quite a bit in projects. Basically, whereas the Human Connectome project is take a whole bunch of people, 1,200 people, and scan them a bit. The NSD is the kind of converse of that. It is take 8 people and scan them a ton. So I don't know offhand exactly how much they got scanned, but it was something like 40 sessions. It wasn't, I don't, well, it's not the biggest data set out there in terms of time each person spent in the scanner, but it's pretty big. And they just viewed, in addition to running like retinotopic mapping and some other basic experiments, including diffusion, they most of that time was spent just viewing like short presentations of color images of natural scenes. That's why it's called the natural scenes data set. And for those of you who aren't in vision, natural scenes, when we say that, we just mean literally any scene that's not like generated by a computer. So like a plaid is not a natural scene, but a picture of a plaid shirt is absolutely a natural scene. So it just kind of anything you could like take with a camera in the real world is a natural scene. So the natural scene data set is great, especially if you want to dig in on visual models. If you're interested in vision, the natural scene data set is a really nice data set. It's been extensively pre-processed. And it also lives up on Amazon S3, so it's very easy for us to access. And by now, this code should be pretty familiar. Like the previous examples, this does not require, like the previous examples excluding the Human Connectome project, this does not require credentials. You can just download it. It lives in the natural scenes data set, which we already saw in my demo. So I'll go ahead and get that started. Now, if we just look at the base directory or the base bucket, there's several different folders, and one of these is raw data. One of them is the stimulus, just what they showed in the scanner. But the main one we're interested in is the NSD data directory. And in particular, there is a subdirectory for the free surfer subjects. I always like to look at the free surfer subjects because I am interested in anatomy. But you can see here that they have each subject has their own free surfer directory. And additionally, they have the FS average and various downsampled FS average directories. Additionally, in the NSD data, there is a directory called PP data, which stands for preprocessed data. And so we can just take an example here for subject 1 and look at the functional 1.8 millimeter data. So this is the data that was collected with 1.8 isotropic, 1.8 millimeter isotropic voxels. So we'll go ahead and look at what's in there. And you can see that there's a variety of files here for various sessions. R2 stands for, it's the fraction of variance squared or the coefficient of determination for the visual models. There's various, some of these are transformation functions, which I'll talk about in a second. There's masks, there's various pre-processed. FLOC stands for functional localizer data. So like they showed pictures of like faces and houses and objects and things, and then localized the fusiform face area and the parahippocampal place area and those kinds of things with the functional localizer. And then eventually we'll get down to, oh boy, there's a lot of these. Well, somewhere hidden in this giant list of files is various like retinotopic data, but we won't worry too much about that at the moment. So I just wanted to give some quick examples here. So let's go ahead and take one of these files, specifically the coefficient of determination or the variance explained for their visual data. And we're again just going to use NeBabel. Here we start with the NSD base path, which is just basically the bucket, the S3 bucket. We go into the NSD data directory and then the preprocessed data directory. And then from there, we go subject 1, functional 1.8 millimeter, and then PRF_R2. So this stands for population receptive field, which is the basic model of how we map out the visual field. And R2, again, is the fraction of variance explained. So what this particular nifty file has in it is an image of the brain at 1.8 millimeter isotropic voxels, in which each voxel has the fraction of the variance explained in the bold signal by the model of vision that they use to model the cortex. So when we load this, it'll be in the shape of a brain. And let's go ahead and just load it. So here we see it's 81 by 104 by 83, which kind of sounds like the size of a functional image. And now that we've loaded it as a nifty, we can just use the imshow function in matplotlib to plot an arbitrary slice. Here I've picked an arbitrary slice, and I'm telling it that since this is actually encoding a percentage of variance explained, I'm going to make the minimum value 0 and the maximum value 100 in the color map. And that looks like this. So if hopefully you can tell this is a brain, and this up here is the frontal cortex, and this back here is the posterior visual cortex. And no big surprise, there's a lot of the variance in the bold signal can be explained using a visual model if you're looking at the visual cortex, but not a lot of it can be explained with a visual model if you're looking at the frontal cortex. So this is just kind of a sanity check of the data. Questions so far? Yeah.
What are the values in the shape?
What are the values in the shape? This is the number of rows, columns, and slices in the nifty file, the nifty image we read in. Yeah. So it's the 3D image of the brain. Yeah. Yep. Other questions? Yeah. What is the range of variance? Oh, like the raw variance, not the variance fraction of variance explained. So that what this is plotting is the fraction of variance. The colors correspond to the fraction of variance explained. Or are you asking what does the variance explained mean for this model?
I guess it's like black is 0 and then white is.
Yeah, sorry. Yes, black is 0. I actually think yellow is pretty much 1 or 100%. And that's not surprising to me. Oftentimes, especially when in a study like this, where you've done a lot of scans, the visual cortex, we can explain the bold signal real well with the models we have. So in like V1, having very close to 100% of the variance explained is not a surprise to me. Yeah. Other questions? Yeah.
So is it just from that visual model?
Yeah, it's a population receptive field model. So if you're interested in that, I'm happy to talk at length about those. But I expect that not everyone here wants to hear all about vision models right now. So there are actually other models included in this data set. So for example, they included an on-off model. And the idea here is that, you know, if you're looking at V1 at the very first cortical visual area, and you flash a stimulus in like the upper right visual field, there's going to be a very particular spot in the ventral left hemisphere that's going to respond. But for higher order visual areas, they may not respond so retinotopically. They may just respond when there's something on the screen. So they have a model that says, is there something on the screen or not? And if that model fits as well as a population receptive field model, that tells you that you probably shouldn't think of this as a retinotopic area. That's an example, but that said, the bold data are also published as part of this data set. So it's all there if you want to run your own models, absolutely. And I believe they've even processed, I believe they've even posted the pre-processed bold data. So if you want denoise data to work with, it's there. Yeah. Other questions? Okay. Okay, now, of course, since NeuroPython is my library, I'm going to show you an example using it. And basically, we're going to import NeuroPython and once again, tell it where the cache directory is. And then basically, we're going to take here. I'm just showing you that this is a free surfer directory, basically. So you can see that if you're familiar with free surfer, it's got the label MRI script stats surf. These are the directories you find in a free surfer subject directory. And then again, NeuroPython just needs to be told the S3 address, and it'll load everything for you and handle it behind the scenes. We just need to ask it for a free surfer subject with this particular address. That'll take a second to load, I think, and then we can go ahead and treat it like an HCP subject. We can just ask for a plot. And this again is going to take a second because it needs to go and download those data. But in a second here, it'll pop up and we'll see the white matter surface for that subject's brain. Yeah. Yes, so they did label like V1, V2, V3. I don't remember offhand how many label areas they labeled, but they at least have those. And then of course, FreeSurfer comes with a certain number of anatomical ROIs built in. They definitely have V1, V2, V3. I think they have HV4. I'm not sure about VO1, VO2 or V3AB or any of those other ones, but and they of course have the functional localizers for like the FFA and some of those. Yeah. Good. Other questions? Okay, just in time, this pops up, and so we have the white matter surface for this subject in the notebook. Okay, other questions? If not, I will hand it back to Ariel for the HBN.
Okay, continuing in our train of different data sets that are all on AWS, the Healthy Brain Network data set. So the Healthy Brain Network data set addresses a whole different set of questions. It's a very large landmark pediatric mental health study run by the Child Mind Institute in New York. It is an ongoing initiative collecting MRI and clinical assessment data from approximately 5,000 New York City area adolescents ages 5 to 21 at 4 different sites. And that's also 4 different MRI scanners along the way. And it includes a broad range of psychiatric, behavioral, cognitive information, including all kinds of interesting biological information. There's really a large range of data that goes along with this. And they published a data descriptor that describes all of that back in 2017. And generally speaking, this data provides sort of a transdiagnostic resource to study development of mental health disorders on one hand, learning disabilities, and just human development in general. A few of the limitations when thinking about next week in the hackathon is that the only information, kind of metadata about the subjects that is publicly available are the age, sex, and handedness of every individual subject. All the data is also openly the MRI data is and EEG data as well as publicly available, but additional sort of assessments like clinician administered KSADs and diagnostic assessments for speech, language, reading, intelligence, and language ability, and so on. All of those are and you know, genetic information, and assiduous baby teeth, and things like that. That you can only access through a data usage agreement, which takes a while to get. So we don't expect you to be able to use that next week. That said, one of the data types provided for HBN is diffusion MRI data. So there's a lot of diffusion data there. There is, I think, several different kinds of MRI acquisitions, including functional MRI acquisitions, anatomical MRI acquisitions. But I'll focus on diffusion for obvious reasons. I find it really interesting. And we've done a lot of work with it. So one of the problems, again, that I mentioned before, processing this data and doing all the analysis takes a really long time, is really cumbersome. In the context of this kind of data set, also, quality control becomes really, really important because children tend to move in the scanner. And so the data is varying quality, hard to say in advance, and hard to derive automatically what the quality is exactly. So what we did in this HBN pod, I don't remember exactly what pod stands for, but it's an acronym. There's open diffusion derivatives in there. I'm not sure what the P is for. Right, this processed open diffusion derivatives project. The goal is to make all the diffusion data available in pre-processed form, but also together with quality control information. So this is a little bit of a diagram that shows what we did. We started off with more than 2,500 scans. Not all the data is, at the time that we started this, not all the data was available. I don't think they have all the full 5,000 subjects yet. So we started with raw data. It's about half the eventual sample. Out of that, we did sort of pretty extensive bids curation of this. And then out of that, we ran, and there were quite a few that were excluded because they didn't have complete data sets that could be bidsified. We ran a pipeline called QSI Prep that does pre-processing. It's a bids app that does pre-processing of diffusion MRI on more than 2,000 subjects. And out of that, we separated away a data set of 200 subjects that a set, I think we were 7 or 8 different experts in diffusion MRI looked at volume by volume to say if they were good or not. So we did QC on these 200. And then we took a subset of that and an additional fairly large subset of the data, and we put it up on a web app called, let's see if we can find it, fiber.dev. This is an idea that Anisha originally originated when she was a postdoc working with me. At the time, we did this on the T1 weighted images from HBN. And this, we evolved this, that was called Brainder. This is fiber. That is that people, anyone from the community could go online and swipe right if they thought that this was good diffusion data, left if they didn't think it was good diffusion data. So we got a lot of scores here. We got a lot of rankings of these data from these more than 1,600 subjects. What we did then is every individual rater was kind of weighted against this subset with the experts. So if one rater was very reliable and also matched the experts very well, we upweighted their contribution to the scores. If they were unreliable and they were just said the opposite of what the experts always said, we downweighted their contributions, create sort of a weighted score for every one of these images. And then we used those images in order to train a neural network that could do automated quality control. And so we did automated quality control of the remaining not so many subjects here. The nice thing about it having now a neural network is that when new data comes along, we can run this neural network on this remaining data as well. So this data set is also accessible through, select, why not? Through AWS, and it's in a slightly strange place inside there under data projects. So it's in this FCP-ND. FCP-ND is actually a big collection of a lot of different data sets. It includes the Abide data set that we looked at also this week, but also HBN. And inside this bids curated derivatives folder, there is a table with the participants. I'm showing you this table with the participants because that's where you will find the subject IDs and scan site IDs, but also where you'll find expert QC score for those 200 that were scored by experts, machine learning based QC score based on the pre-processing pipeline, and then this deep learning QC score. So in addition to, if you're interested in denoising, quality control, things of that sort, this is a neat data set with lots of ground truth quality control, but also a variety of different other quality control scores for every subject. In addition to that, there's all the tractometry stuff from PyFQ in there. So here's, the FA from the corticospinal tract, left corticospinal tract of one of the subjects there. Very similar to that data from the Human Connectome project in terms of layout and the bundles we extracted and so on. So that's the Healthy Brain Network. What questions do you have about that? Cool. All right. On to the mega data set.
So now the catch-all. Okay, so I think Russ mentioned OpenNeuro in his talks. So OpenNeuro is this great website. Basically, if you have a data set in bids, they will take it. And they'll store it there, and you can make it accessible. And so I highly recommend any data you want to publish, put it in bids and send it to OpenNeuro. It needs to be neuro data, but yeah, it's a great place to find data sets. And I'm going to start by just going to the website, openneuro.org, and make this a little bigger. But you can just search. So being a vision person, I'm going to search for retinotopy. And the first data set over here is the NYU retinotopy data set. So I'm just going to click in on there. And there's a little information about the data set here. You can actually browse the data set in their browser here, and you can see that it's got it's a BIDS data set, so it's got derivatives and it's got raw data for all the subjects. I think this has about 50 subjects. I'm not sure offhand. Information about metadata and what's in the derivatives. You can just download it if you want, or it's got instructions on how to download it. But what I wanted to point out here was that when we came over to this site, it brought us to, and I know this is really small, but it brought us to openneuro.org/datasets/DS003787. DS003787 is the ID for this particular data set. I believe it's also on this page somewhere. Right here. The Open Neuro accession number. So 003787. So if we want to now access this data set, you can see that I planned this out a little bit. We just give the S3 path S3://openneuro.org/ and then the data set ID. So in this case, just /DS003787. Again, this one doesn't need credentials. Open Neuro is anyone can access it without logging in. So I'm going to go ahead and run that, and then we'll just do an LS on the base path, and you can see basically the same thing we were just looking at in the browser, the derivatives directory and a bunch of subject directories. We can of course read in the use pandas to read in the participants.tsv file, which we kind of already saw. And we get back the participants as a nice little pandas data frame. And then once more, I was just going to demonstrate how to load these into FreeSurfer. And it's pretty much exactly like we did last time. We just, load these into NeuroPython. We asked NeuroPython to give us a FreeSurfer subject with this particular path. And again, it'll handle everything. I picked that particular subject because that's me. From back when I was at NYU. So in a minute here, we'll see my brain, or a rough model of it. But one of the great things about Open Neuro is that since literally like anything is on there, and anyone can put data there, there's all kinds of data there, and you can just go search and see what you can find. Lots and lots of labs have put data on that website. Any questions while this loads? All right, well, it will load in a second.
How much will you pay
Nothing. It wasn't until after I left they started paying grad students and postdocs for experiments at NYU. And there we have it. All right. So I think this is oh, is there another question? Oh yeah, great.
Yeah, there's one more data.
Yeah, excellent.
Actually, it's not a new data set, it's also open.
So I do. I can. You want me to go?
If you go to query.neurolego.org,
Neuro. Okay.
So that's, I think, is not completely ready for consumption. That's annotation at the sample level of all the open neuro data sets, such that you can say do like minimum age of 70, for instance, and then do sex equal female, and then query that. So we do a T1 modality, whatever, and it would actually pick up in all those data sets all the females that are older than 70 years old. And so you can then select all maybe the data sets and then download a CSV file where you have all those variables. And the thing that is not entirely ready, that's why I think it's a bit premature for this group, but it's hopefully will happen soon, that we have the the data lab link to the T1 image that you may need to you want, or whatever the modality you've chosen. So that's a cohort creator rather than a dataset finder if you want. And that's eventually you will have different endpoints with more than open layer. Yeah, so that's it.
Oh, thank you. And this actually reminds me, obviously there was a little bit of bias in the data sets we showed you because I'm a vision person and Ariel is a diffusion person. And so we kind of focused on those. But Open Neuro is sort of our catch-all here. There's every kind of data in Open Neuro. So if you're looking for like an EEG data set or an MEG data set, those exist in Open Neuro and you just need to go look for them. And if you're looking for other data, we're happy to help you as much as we can. We're certainly willing to work on data sets that aren't vision and diffusion. We just maybe don't demo them. All right, any other questions? Okay, I think we can break then, and we'll come back after the coffee break for review sessions. I believe that here will be Ariel and John, and in Alder 103 will be me and Kelly. And at least in Alder 103, Kelly's going to start out for the first half hour to hour, however long it takes, talking about fMRI prep specifically, because some people asked about that. So we'll be starting with fMRI prep over there, but it's the review session. Feel free to move around between the rooms as you feel appropriate. And we're just going to be here to answer questions, basically. Yeah.
So you're going to talk about your classes?
Yes, or whatever else you want to ask us about. So we're definitely prepared to answer questions about anything we taught, but also anything we talked about in general, or any other questions that seem related to what we're doing is totally fine.